Hello, everyone, and welcome to Office Hours Live with the Meter Environment team. Today's session will focus on soil moisture, and we're shooting for about an hour of live Q&A with our experts, Dr. Colin Campbell and Chris Chambers, whom I will introduce in just a moment. But before we start, we've got a couple of housekeeping items. So we first want this session to be interactive, so we encourage you to submit any and all questions in the questions pane, and we'll try to get to all of your questions. I'm just going to say here right up front, we will not get to all of your questions. More than likely, only a few of them will be answered during this hour, but we will have someone from our science and support team get back to you with an answer via email or the email that you registered with. So second, if you want us to go back or repeat something you missed, don't worry. We will be emailing you a recording of the session via your email within the next 3 to 5 business days. All right, with all of that out of the way, let's get started. Today, our panelists are research scientist Dr. Colin Campbell and application specialist Chris Chambers. Colin Campbell has been a research scientist at Meter for 20 years following his PhD at Texas A&M University in soil physics. He's currently serving as Vice President of Meter Environment, and he's also adjunct faculty with the Department of Crop and Soil Sciences at Washington State University, where he co-teaches environmental biophysics. Dr. Campbell's early research focused on field-scale measurements of CO2 and water vapor flux, but has shifted toward moisture and heat flow instrumentation for the soil-plant atmosphere continuum. Chris Chambers operates as the support manager at Meter Group, and he specializes in ecology and plant physiology and has over 13 years of experience helping researchers measure the soil-plant atmosphere continuum. So thanks for joining us, and let's get started taking some questions. Thank you to everybody who's sent in questions. We sent out an email to register for this with an invite to submit questions. We've got a ton of questions. So like I said, we're going to try to hit some of the questions that came in via email, and we will also try to get some of your live questions. So feel free to submit any and all questions there in the questions pane, and we'll try to get to as many as we can during this hour. So our first one here is, we're going to start here. This questioner asks, in my experience, field capacity varies in terms of KPA among different soils. Would you agree?
Thanks, Brad.
Yeah, thanks.
Good morning, Colin.
Hey, good to see you, Chris.
Good to see you too. So field capacity, this is a question that we actually get all the time, and it's appropriate to hit permanent wilting point while we're talking about this. Let's start by just a quick definition of field capacity. For those of you who are new to the topic, it's basically the point after a soil hits saturation where all of the gravitational water drains out. And so if you have a bucket full of saturated soil, a bunch of holes in the bottom, you hit field capacity after there's no more water draining out. Just a quick definition, right?
Yeah, and some people call it the drained upper limit. Other people say it's the time when no longer does vertical flow dominate. So lateral flow and vertical flow are about the same. And depends on your level if wanting to talk about it. But in my soil physics class or even other classes I've had, they all said, oh, it's negative 33 kPa.
Right. And we see that quite often. And that's really attractive from one point of view, right? Because it's always the same, in the definition I just gave you. That's super subjective. Right?
But I didn't know that when we first learned about it, right? That it was like, hey, here's a number you can depend on, negative 33 kPa. And the interesting thing was that we've started using our Teros 21 water potential sensor. I've always known about water potential. In fact, grew up really knowing about it because my father worked so heavily in it. And suddenly, now we measure that in the field, and it's not negative 33.
It's not always negative 33, is it? That's the answer to your question is, yeah, that's pretty much right. The field capacity can have a different matric potential or a different potential depending on the soil type. And
so what would you, you've seen this before in support experiences. I think I remember one of the funniest moments. We sell a WP4C, right?
Yep.
Somebody wrote me an email and they were like, well, I know your instrument doesn't work because I put some well-drained soil in the WP4C and it didn't read negative 33 kPa. One of the problems was the instrument simply doesn't really work in that really wet region.
That's pretty wet.
But the other problem is that's a fallacy. But it shouldn't have.
Right.
So sand, clay, what do you expect to see when you go to the field?
So for me, and this number is really important, right? Because but it's kind of a physical construct. That it's an important conceptual point in the soil that it's good to understand the soil physics and how water moves through it. But I find that less interesting now that we can measure matric potential a lot better, especially with the Terrace 21. And with water content and matric potential together, I just think that we're kind of stuck in the rut of what water content or what matric potential field capacity is, because now we can measure it on a continuous scale in the field.
I get what you're saying, and that's a concept I hadn't thought about too deeply. At one time, we were trying to understand field capacity because we were measuring water content often.
Right.
And so if we could say, okay, after 2 days, that's kind of field capacity, and that's maybe around where the plants grow optimally, we want to nail that value each time. But if you're using a Terrace 21 or even a Terrace 32, a tensiometer, maybe for more accuracy up in the wet range, you don't really care because you know the range.
That's right. I don't think so because, as we said, it is an important physical concept, but in the end, the definition of it is kind of subjective, and we don't need to rely on that subjectivity to find out the soil moisture state.
But what if you really want to know the field capacity of a sand?
Oh my gosh!
I mean, in my experience, maybe
right.
negative 10 kPa. So if we define a point,
you're right. It is important because that's where your refill point is. Right?
Yeah, and that's where we're going to. I mean, anywhere above that, if we put more water in, we're essentially donating to the groundwater, right? And we're doing that with our nutrients, with whatever's in the soil, we're sending it by. Sometimes that's important if we've got solid affected soil. Maybe we'll get to a question like that later on. But here's an interesting thing. In the turf grass we're working in, this is a pretty sandy soil. Obviously, the whole point is to drain. Negative 10 kPa is about what I expect to see. I looked at some of those data this week. When we installed in a potato field down in southern Idaho, what we saw there was negative 40 to negative 60. And that was after a nice winter, good snowpack. This was high elevation, and it's all melted and it's just sitting there. Nothing's growing on it. They're getting ready to plant. Negative 40 to 60 kPa. And that was their field capacity, essentially what moisture is left there after it's drained.
Yeah, right.
So, okay, so this question didn't ask this, but what about permanent wilting?
Those go together, right?
They are. And the permanent wilting point is something we even know maybe even better than our field capacity.
Well,
I mean, in terms of numbers.
And this comes down to, another important physical parameter for the soil. And just, it was originally determined, I can't even remember.
1910.
Yeah.
And Briggs and Shantz, I think.
Right.
You can go look up the paper.
But essentially, the permanent wilting point is defined as the potential, the matric potential at which sunflowers permanently wilt. Right?
Yeah, so they fail to recover after the night of, coming across.
Right, and that's that minus 15 bar, that minus 1500 kPa number has just kind of been the standard ever since. It's objective, it's super easy, but it is going to be different from plant to plant. And
why is that? I mean, you talked about that earlier, and I loved kind of.
So you think about a sunflower, and they grow extremely fast, right? High assimilation rates, and they use a lot of water during that. But different plants have different strategies to avoid plant stress or to protect themselves from plant stress. Say a conifer, a ponderosa pine or something, they might get down to minus 2.2 megapascals before they really have, they really enter a stress zone. And something like that with woody tissues, really sclerified, but I mean leaf needles that are really hard. The wilting isn't really the right terminology for that either.
Well, and that is interesting. Right? So the needles, I mean, they exhibit a different physiology that maybe my potato plants. In terms in facing stress.
So, but then again, you know, as we get the actual matric potential, then we can not just kind of benchmark this one point, but you know, look at the data as a continuous variable. Yeah,
so we've done some of this work. You know, what is wilting point anyway? Why are we interested in wilting point? It really, you know, the answer to that is that wilting point is something we want to keep our plants in an optimal range, and wilting point is something we never want to approach. Right. Interestingly, it's manifest. You know, when I work in wheat, when I work in, you know, other just baseous plants, when I work in potatoes, humorously in potatoes, they're the ones that are like, no, there's no water here, we're giving up. You know, and then they run that white flag out and they're like, we're done. You know, and wilting point is just something you can never approach because it, you know, for something we're trying to produce yield out of there, wilting point would be crazy to reach.
That's right.
But in wheat, you know, you give wheat a challenge like, hey, you've run out of water, they just send their roots deeper. You know, and when we did some measurements here locally, we had water being taken up at 150 centimeters, just like that toward the end of the summer. They're like, well, we'll just grow roots down and grab it. And so wilting point, important parameter, not negative 1500 kPa, right?
Right. Or maybe it is.
Well, who knows?
But you know, it does give us kind of an objective way to define the water tank.
So, so we'll go there, but let's not. Don't send me an mean email about how the WP4 doesn't measure wilting point. That same person actually said that on the wilting point side too. Okay, Brad. Another question.
This individual is asking: does sensor orientation matter, and how many would you recommend installing in a field scale study?
Yes, it matters, and lots.
And no, it doesn't. The sensor doesn't care. If you can install it up or down, vertical or horizontal, and it doesn't care.
Oh, that's true.
But it does matter on, you know, how water moves through the soil around the sensor, how you're installed, what you're doing with your cables. Cable management is always important in installation, and installation is, if you pay attention to nothing else in your when you're installing soil moisture sensors or when you're doing a soil moisture experiment, how you install the sensors is the single most important thing you can do to get good data.
And so what can we give as a principle as people going out of the field? Hey, if you remember nothing, remember this. And if it was me, after all the many installations, what I would say is water gets everywhere, and really take care of that. Don't impede the water flow by laying your sensor flat, right?
Don't aid the water flow by having your sensor cables have a channel for water to get straight to your sensor.
Right. And one of our good friends, Georg van Unold, talked about how he installed his tensiometers at 45 degrees, try to limit the preferential flow. And I think, you know, in every learning I've done with water content sensors over the years, or anything installed in the soil, again, it's water will get to the most bizarre places. It will get inside your sensor if you're not careful. It will get down. It will flow down your install hole if you're not careful. It will do everything you don't want it to. So,
what about the second part of this question? You want to tackle that one?
Yeah. So I think you should. Like 80 or 90 sensors across the field.
If you're not installing 80 sensors, you're doing it wrong.
Yeah, I think so. I think you should spend all your money on sensors, all your budget. No, in all honesty, this question is really challenging. It does relate to budget, right? I mean, there's always a finite budget. And we've done this a lot of different ways. I mentioned the study on the wheat a little earlier. We installed 42 sites in a 35 hectare field outside of town. So that's a really dense sensor network. And we discovered some really interesting things. In fact, one of my data science friends who got a PhD in geography said, hey, that's one of the seminal data sets people are using right now to do some of the analysis out there. And so you can do that, but I don't think a lot of budgets are going to be able to handle that, and it takes a lot of time. It took us a bunch of time.
Heavy analysis too.
I mean, you had a good idea on this.
I fall back to just your basic statistical sampling principles. One, you want to be representative of your overall population that you're sampling, right? If you're placing a sensor in a field, you want to be representative of the entire field. And part of that is going to be dependent on the soil variability in your field. And then second, you want to get a handle on the variability by having kind of at least the minimum sampling number.
Yep.
So per soil type, you want to try and have at least 3 sensors in there, which is the minimum you need to calculate a variance.
Yep.
So, if you have one soil type and it's consistent throughout the entire field, you can, which I think is fairly rare in many fields.
I've never actually seen that happen, but we have installed that way before because that was just our limitation.
And sometimes that's the best that you can do. So finding the spot that's representative, the most representative of your field is it's tricky.
It is, and we've been trying to deal with that some. We've put together a little app that will take satellite data and try to create statistically representative locations in the field and give you a suggestion of where you might put that. We look at NDVI, normalized difference water index, and some of these things and overlay it and say, hey, this might be a great spot to put sensors. Right. And so that's so far our best guess. I think the future looks like we're going to spend a lot more time trying to connect all these scales. I mean, that's what everybody's talking about.
That's where we're going.
Okay, Brad, let's try another one.
Along with orientation, this one's asking, where does the sensor have to be? In the most active root section, in the center, at 50% effective root depth, and also where are you going to do that in a drip irrigation situation?
Ooh.
They threw in that curveball, right?
Yeah, the drip. This is really related to the question that we just had. And it depends a lot on the questions that you're answering. And what other resources that you want to use. Certain soil moisture networks, SNOTEL sites, a lot of the mesonets that provide soil moisture data as a product, they have set depths. So if you want to compare and interact with those, if that's one of your questions, then you should use the same depths that they're using.
But they don't always agree, right? I've been to meetings where they're
that's
like,
right.
No, it's 20 centimeters. No, I think it's 25.
It's exactly. And then, but if you're really interested in what water is available to your plants, then you want your sensor to be in the root zone.
Yeah, I mean, it's something we've talked about, like the potato example I used. So we've been going kind of at mid-level, upper to mid, and then at lower, toward the bottom of the root zone. And then I've been sticking a sensor out the bottom, out the bottom of the root zone to see
what's getting through, how deep the roots are reaching.
Yeah. And then the question is, well, what sensors are you putting out there, right?
Yeah.
Right now I'm co-locating water potential and water content sensors. I want to see, essentially trying to create this envelope, right? Of upper and lower limits for water potential and what that looks like in water content, and therefore how much water to put on.
Well, and that gives you information about your soil too, and what's happening, what soil type you have, what your soil water retention is, and
it's a fingerprint for the soil. That's what I've been excited about doing putting these sensors in.
You get the mass and the energy state at the
same time.
Yeah.
So that's where I've been installing. I like your answer, though, about these networks, because there are kind of standardized ways to look at this. As we've worked with growers, though, I have my own thing. I was like, okay, we're going to put them at 15, 45 centimeters. And the grower basically said, I'm putting him at 30. And you know what? Guess where we put him? At 30.
Yeah.
He didn't care. So drip irrigation.
Oh, my gosh. Okay. I think you get information from, again, you probably want it in the root zone.
Yep.
And you probably want to know the extremes. You want one near the drip line and you want one away from the drip line. especially if you have plants that aren't right on the drip line.
Yep.
And if you have the drip line running right down the center of a row or something like that, then you might not care what's going on in the interrow.
We had this experience last summer. So we're in a silt loam. We have the drip line going down through. Okay, and we have a big inner row, so they're 2 meter spacing. And so the question was, where do we put it? And so my thought was, what we could do? We could go out and just look online. Why? Yeah, terrible. In fact, I was talking to our friend Doug Kobos, and he was like, "Hey, that's going to be simple. Where to put this underneath the drip line? We'll just go look in the literature." Now, I'm not going to say I did an exhaustive study. I asked, Captain Google or whatever, right? And so I went to Google Scholar. I went around to these things, and it was like, okay, where do I put my sensor? There was almost nothing. The best I did was like, hey, put it 10 centimeters from the dripper, laterally, and 10 centimeters horizontally from the dripper. Go there. And that's what I used, and I can't tell you if it worked. Because, we measured, under the drip line. We got the onion, and it was certainly interesting, but was that the right place? I don't know.
How far does it go?
Yeah, so
far does it
there's a potential study. Maybe I've just missed it, need to spend more time, but that's what I got.
We've got one that says, when measuring soil moisture at varying depths, should the sensors be vertically stacked for different depths, or is horizontal spacing also recommended? And if so, at what distances?
They're both important. Both, you want some laterally to capture the soil variability. We've already touched on that a little bit, but water moves vertically, and there's a lot of spatial variability for water laterally and vertically.
Sure. Flows in fingers. People don't really realize that, but it's not like a big block coming down through the soil.
That's right. And again, this partly depends on the questions that you're asking. Sometimes knowing what water is if it's below your root zone and that water isn't being accessed, if you have a reservoir down there, that can be helpful, or what kind of drainage you're getting. To have that deeper sensor can really give you a lot more information.
I don't think I'd go laterally, and here's why. Borehole installation tool.
Oh, yeah.
And I have to admit, I have put in, I don't know, at least several hundred, probably more than 1,000 sensors in the soil in various installations over the last many years. And when I got a hold of the borehole installation tool, life changed. Not for the water potential yet. We're working on that. But for the water content, digging that hole, even in rocky soil, and then using the borehole installation tool to just shoot those sensors into the side of that borehole changed my life. And that sounds Homer, right? Because we build that, but I love it.
Well, we had that trial too, where it's like, we're going to get our whole meter environment staff out and we're going to dig pits. And so we tested it. We were digging. I was the control group. I led our team of interns to dig the traditional pit. And you and Leo were done. Had your sensors installed and your holes cleared up in 30 minutes.
That's right.
And we're still digging.
Yeah.
Digging down to get to.
What did we do with that video?
1.8. I don't know.
That'd be a fun one to connect. So
we're digressing.
Yeah. Essentially what I'd say, we've done this different ways before, and we did in that field, the 42 site one. Remember, we did poke holes kind of laterally. Yeah. Just makes me a little nervous based on soil water flow that we may miss. If you're stacked vertically, at least you got some idea that the water's flowing down through a regular pattern.
That's right. I still think the lateral, especially if you have a lot of soil variability, because you can have like a pretty significant soil texture change, and your soil moisture they can both be right. They could be 15% off. Just
it can't be away. Absolutely.
So I still think the lateral is important. Hopefully we gave you guys something to think about on this one.
That's a hard one. Okay, Brad, what do we got?
How does salinity, temperature, and capillary rise affect the data from the sensors? Is there a threshold for the noise it creates in the data?
Brad's throwing us some pretty hard ones.
This is why we're here, though, right?
Yeah, okay. Yeah. Okay, Chris, why you're here. I just want an easy morning.
Salinity. Let's start with salinity.
Yeah, great. So we've spent a long time worrying about salinity measurements from the sensors. And one of the challenges with measuring the charge storing capacity of the soil through a sensor like the Teros 12, Teros 10, 11, 12, is that the charge storing is affected by the salinity. So we've spent a lot of time testing the sensors, and within a reasonable range of salinities for most soils, there's not a lot of effect of those things, of the salinity. But, if we get higher salinity values, maybe above 10 decimum per meter.
Definitely above 10. I'm pretty sketched out. Saturation extract EC.
Yeah.
So if you saturate the soil, collect the EC that's coming out the bottom of it, and it's above 10 decisiemens per meter, then like the Teros 12, your data, you'll probably see it if it's affecting your data. It's pretty clear response. And especially if you're measuring EC, then you can have an idea of when to just maybe not completely trust the data that you're getting. But
so how do you know what the EC salinity is?
So that's right, because
a soil sample, maybe?
A soil sample, generally you can tell from in the soil. A bulk EC, if your bulk EC is above 3 to 4, that's a warning sign.
You've got problems. Depending on your water cut. If your water cut is low, you're in real trouble.
And so if you're measuring EC, you generally have indications of when your water content is going to be affected. But that's a pretty high EC level at 10 decisiemens.
It is. I've dug into the salinity labs at Riverside produced a handbook many years ago, handbook 60 maybe.
They have some where it affects the sensor.
Yes. So, but they've showed that certain crops growing. A saturated extract EC of 10, that's getting into only a few crops are going to be able to grow in there. So it is a concern. Okay, capillary rise.
That's just fun. Hopefully you've got matric potential sensors in there at 2 depths, because then you know what's happening.
Yeah,
right.
Coming up from the bottom. Yeah, I've never.
And we see it occasionally. And
have you dealt with that? I don't think I have.
Yep. And so in the older sensors, this partly depends on the sensors too. In the MPS 6s had a little more temperature.
Yeah.
A little more temperature effects in them, and it was sometimes kind of hard to see the difference between a diurnal temperature effect and hydraulic distribution in the soil.
Okay.
But I don't really see it very often, or we don't see it clearly very often. There's a handful of cases where, this looks like.
Yeah, that's pretty rare to see some of these things. The temperature at a very, very dry in the Teros 21 in the water potential, we can see temperature effects, but then again, temperature does affect also the water potential.
Yep, that's right.
And in fact, I was just kind of bemoaning that fact in a Teros 21 I was looking at, and also had a tensiometer, which I trust is kind of the golden, the kind of thing, the standard you look at, and it was the same thing. There was temperature effects almost identical in that sensor, which really suggests it was something related to
the physics,
right, the physics of the soil. So, okay, let's try another one, Brad.
Is there a way to calibrate the moisture sensor for soils of different textures and different bulk density? Can you correct a moisture sensor reading?
Yes. Next question.
We should spend a minute on calibrations.
Okay, let's do it.
Because it does come up, it is relevant. With our factory calibrations in a mineral soil, and we'll focus mostly on mineral here, generally they'll get within plus or minus 0.03 meters cubed per meters cubed. Mm-hmm. And for a lot of applications, that's a custom calibration isn't worth it. It is an investment in time and effort to do a custom calibration. But if you have the soil sample and your soil moisture readings, you've got a season's worth of data that just doesn't look quite right, then you can go back and correct your water content by doing a custom calibration there.
So too regularly people ask me, how do you do this calibration? And we have an application.
Yep, it's our procedures online. It is pretty straightforward. It does take some labor, though.
It does take some work. we have lab technicians who scrub the little clay balls, wet clay balls, try to homogenize the soil. I used to do these a lot.
Yeah.
But one of the good things is it creates really good calibrations. And I've seen a lot of people like, you know what we're going to do? We're going to skip all that work. We're going to make a little tray. We're going to put the sensor in the tray, put the tray on the scale, and we'll just let it evaporate.
Right.
And it creates terrible
That's right. It is better to just use the factory calibration than to not do the calibration correctly.
Yeah. I'm glad you said that because I hadn't really thought about that. If you're going to do it badly, just use what we did, because I can tell you that we spent time on it. But if you're going to take your time, definitely calibrate if you want to.
You can get a 1 to 2% improvement, and some soils do really need a calibration.
Yeah, absolutely.
We've had some come in with crazy bulk densities.
So we were talking about this earlier. Talk about the bulk density. What range do we need to worry about?
Generally,
what is the typical bulk density?
Generally, the sensor is pretty robust to bulk densities for mineral soil, and that's from like 1.25 to 1.7.
Yep.
You're anywhere in that range is probably not a big enough difference in bulk density to require a calibration.
Yeah, I'd agree.
And you probably have soil type differences in between there that are, across that range as well. Where we've really seen crazy data is, I don't even know, the soil came from somewhere in California, but it came in with like a bulk density of 0.9 grams per centimeter cubed.
Could you tell?
And
just when you
I couldn't tell, but the water content readings were crazy. And so that definitely needed custom calibration for that one.
And so, we also, in that calibration sheet, if you just want to take the bulk density, there's ways to calculate that in there too.
Yep, exactly.
So that's what I'd suggest too. Volcanic soils, for example, need it. Okay, Brad.
Asking about their application here. So I'm just going to paraphrase because it's a long question, but they're saying that they're growing 1-year-old cork oak seedlings in 5-liter pots. They're saturated, dried down. They did an experiment measuring the pre-dawn leaf water potential and matric potential in the root zone and did not get a very good agreement between the two measurements. Can you explain why they would see a difference?
A pre-dawn water potential is a pretty common tool for physiologists to understand the basically it's kind of a hack to get at the soil matric potential by measuring the water potential of the leaves.
It's a hack because you have to get up at pre-dawn. This time of year it's like 3:30 in the morning.
But sometimes
so it must be valuable if you have to get up that early.
It's hard. If you don't have to do it very often, it's fine, right? Like
I've never done it.
Like
I'm gonna admit right
watching
here.
the sunrise at the top of Smoot Hill.
Right, okay.
Because you've been up all night taking your readings. Not all night, but anyway. The point is that in theory, you can get the matric potential of the soil by measuring the leaf potential. And you can do that because when the stomata close, the plant and soil come into equilibrium.
Yeah.
And that's the key assumption that's happening. It's really hard. I think it's really hard to validate that measurement, to know whether you reach that equilibrium. It's also really hard to tell where in the soil that particular leaf is connected with.
Yeah.
Right.
So this is the traditional wisdom, just as you say. Like in class, you learned that, I learned that. Overnight, pre-dawn should kind of relax the transpiration stream, and we should all be in agreement. And this particular question asked about 5-liter pots. And planting trees, they saturate the soil and just let them dry down.
Which you think would be the ideal condition to test this, right?
I like it.
A small soil volume, a pretty small canopy.
Yep.
There's only so many places you can connect it there.
Right. There are a couple questions I have on this that I didn't see in the question, and maybe I didn't read quite carefully enough. But one was what soil they actually were using. Now, our friend Leo Rivera did some studies on some of the potting soil mix and found that hydraulic conductivity does make a big difference in that. I think that's what you were getting at. It's like, where is the plant taking that water that's then in this leaf? And if we have a coarser mix, some sunshine mix, as all the universities like to use, the hydraulic conductivity gets lower. So we might be measuring very high water potentials at one location, meaning close to zero, very wet, and yet right next to the root, the water flow is poor, and so the root can't get at that water. So that was one possible reason.
Yeah, it's from my point of view, this is a really hard question to answer. It's just so tricky to validate what's happening in the.
And I did some work last summer. I actually had the same idea, but I didn't have this great idea of doing it with oak in pots. I went out to an apple tree and a pear tree, and I measured the stem water potential continuously with a new sensor that's out there, and then some soil water potentials. They didn't agree either. And I felt maybe this at the heart of this question, like, what is going on? There is a gradient, though. So certainly during the nighttime, we assume the gradient. At night, we assume there wouldn't be so much of a gradient. But those assumptions are made because stomate's closed. We think everything kind of relaxes in there. Right. For sure, the daytime water potentials in those leaves, in the plants I was measuring, for sure that was tied to atmospheric demand. I could match ET up with leaf water potential or stem water potential, and those were highly correlated. Nice. The pre-dawn water potentials, I didn't go measure. I tried to make the technicians to go measure, but they told me no. So I don't even know I don't know what was going on there. So the answer to that is, so what's the answer? I actually looked. They were an order of magnitude different. Negative like 100 to 300 were the soil water potentials, and negative, let's say 1100 to 1500 were the leaf water potentials.
That's a big difference.
Yeah. Any other reason why that might be?
I'm not sure. It's easy to say, well, trust the sensor because the sensor's right. It's calibrated. It's normalized. You can get that out and test it.
I'm confident in that reading.
That's right. So that reading is good. But again, there's things that we can't see coming through. It sounds like you're looking for me to just definitively answer the question, which I'm not going to do.
Come on,
right now.
Chris.
Nope.
You're gonna shy away from that.
I think this one we can have a lively discussion, but in the end, I don't.
I think more research. That I'm confident. I've used the Scholander pressure bomb pressure chamber for years and years. Yeah. I imagine even if we had been testing with some of these new devices, we would have seen the same thing. Maybe not as large a difference. I don't know, but I think it has to do with the physiology. I also think there's a unsaturated hydraulic conductivity in the soil potential issue there. Mm-hmm. If it was native soil, then that would take it away somewhat, but that's what I answer so far.
Right.
Okay, pick an easy one, Brad. We're all tapped out. I'm just kidding.
How about let's go back to field capacity. All right, so they're asking, is field capacity or does field capacity in one particular texture change during a cropping season? I guess this could be, throughout any season as well, right?
I don't know the answer to that.
So last summer, I was out working in potatoes, and I gave, these growers, I'm like, okay, here's the range. We need to be about, let's say, negative 20 to negative 60. Mm-hmm. And these one growers were at like negative 120. And I'm like, man, you guys are super low. And he's like, you need to recalibrate your sensors.
Oh, no.
And I'm like, oh, no. I mean, yeah, it's possible that a single sensor could be, for whatever reason, not behaving. But we had multiple sensors all telling us the same thing. Yeah. And in the end, I'm like, no, we can't. You need to recalibrate your water potential scale. I'm like, I didn't make the water potential scale. Plants made the water potential scale. So I can't do. We can't do that.
That's sometimes a jump to move from a subjective method of doing things, and then to get this objective.
Yeah, exactly.
This objective thing that's is going to skip the biases and other things, and it might not provide the complete picture, and it has some uncertainty around the measurement, but it's going to give you objective information to work into your decision making.
That's the take home, right? I'm trying to say, look, here are some standards to live by. If you think your plant, is happier at negative 120 kPa, which I actually don't think, but if you think that's true, that's fine. We can say, oh, this happens to be a variety of potatoes that's super good at negative 120. Maybe it's because we have a problem with this, center pivot, the sprinkler system. But I guess that to get back to this question, is field capacity, does it change? I would say no. I would say maybe a little bit, like here's one case where it might.
Structure.
Yeah. So if we have roots growing like in a turf grass, in a sand, if we had a change in structure, quote unquote, where we have a lot of roots now filling those small gaps, the fine roots are filling the gaps in the sand, then we have it. We could change our moisture release curve, our field capacity, that kind of stuff. All things being equal, in what I'm more used to, in a kind of a, even a sandy loam or a silt loam or something, I would say
it's generally a soil property.
It's just a soil property we can count on. Although I will say that when we look at in situ moisture release curves from Terros 10, 11, 12, and a Terros 21, we do see a little bit of differences over in the season that may change a little bit. And I don't know why
that do you think is from installation?
I wonder that. I don't know. The often in a lot of these irrigated soils, we have to take sensors out of the season, and some of that might be settling. We're just starting on, for example, our second year in some of these turf grass applications where we have let them overwinter. Data are really interesting this year. They changed almost on their head from what we did last year. So maybe one of these I'll try to answer that question.
Great.
Okay, Brad.
Similar, asking about field capacity and permanent wilting point. So given this specific topic, how useful or relevant is soil water content tool or model, in your opinion, to use a reference for field capacity and permanent wilting point?
We're there.
No,
we're there.
Here's what I would say. Here's what I actually thought you were going on this one. Let me just say, I just read an article about some of this stuff. It was actually an article on the information gap and water potential in the field. Right. And they were talking about pedotransfer functions and saying, well, look, a lot of you guys use these models, these pedotransfer functions, put them in your models and are estimating the field scale or even larger scale, catchment scale, movement of water, the water balance, et cetera. But these pedotransfer functions, because we don't have a lot of information on the water potential, they're in big error. And so what I would say is, if you just go out and use a reference field capacity permanent wilting point, if you drive some of your understanding just by go and get the Sergo data, get your soil type, generate a pedotransfer function, and you think you're good to go, I would say no. I think that the data would suggest that understanding what's going on in the soil better, improving our measurements, filling the gaps as the kind of the clarion call goes right now. Fill the gaps. We just don't have enough field measurements. I think that's important.
And they are important benchmarks, but they're maybe a little bit more. They were a little bit more helpful when they were the best tools we have, and now we have some tools to.
I love that.
To help move beyond them.
Yeah, I love that concept.
That's the train of thought. We're back on the rails here.
No, that's great. We're heading down the track. No, I agree. But I get stuck in that trap too sometimes. Honestly, yeah. I mean, I've been teaching environmental biophysics for 22 years now. Right. We do it out of a book that was written, and released in 1998. There is new knowledge out there. That's one of the reasons I love to bring students into the class, is because they often teach me things. And one thing to think about for me is that I sometimes reject their new knowledge, shall we say, just because of my kind of age bias. I come in and I'm like, hey, I already learned this. My teachers taught me back this back in the 90s when I was taking classes. We're good to go. And really, we've got to embrace the tools, the new steps that we have. It's hard for me, but there's a lot of learning we can do. Right. So I would be careful of that. That's, I guess, my end point.
This next one, we're going to go back to talking about salinity. So what is the relationship between salinity in soil and available water?
Looking at this from a sensor perspective, which is kind of my default position, right?
Okay.
Taking the measurements.
Yeah.
We measure the water content, right? We measure the EC, and those two things, the water content, the EC is dependent on the water content to a certain extent, right? You have the water content and the solute concentration of your solution that are going to be the main components of the EC of the soil. And the bulk electrical conductivity just isn't always that helpful, right? Because unless you can control for the water content, you have two variables in the bulk conductivity that you're trying to, and you can see one of them, the water content, independently. And so we use some EC models to kind of try and help us get at this. And so for the water content, the available water is generally independent, right? But the EC is kind of part of the
Let me ask you a question, though.
Go ahead.
In terms of salinity, have you ever experienced a salinity that actually caused a reduction in available water?
Well, that's from the plant perspective.
Yeah.
Once you start hitting plant stress.
It's interesting. That's where I went with this question. You went in the limitation water content, which is also a key piece. But I got it running down this road of the matric potential versus osmotic potential.
Right.
And we have been doing quite a bit of work in the managed ecosystems in horticulture right now, talking about the osmotic water potential. And something I didn't even know. I didn't even. So there was years ago. You were a young employee at Decagon Meter Group.
You're like a year older than I
am. You were just recently returned from your vagabond test.
Okay, that's a better way to.
And you walk in, and we were testing this new MPS-1, our original water potential sensor. Do you remember that?
That's right.
And we put it in a potted plant, and we're like, we'll be able to stress our potted plant and make it do all these great things, and nothing happened. Mostly because of this saturated, unsaturated hydraulic conductivity deal. But there was also another thing that I didn't even know, and I don't know if you knew, that really, to pull water all the way down in this potted plant to the point that it would be stressed, couldn't get the water anyway.
Yeah.
And the only way to stress this would be osmotically. And it turns out, I've always thought that osmotic stress in plants would produce a salt toxicity. And so far in the work we've done, which is not on a lot of plants, the osmotic stress that we can create with this actually doesn't cause salt toxicity, and it actually does cause the plants to flower more and put more energy into their fruit.
Oh, nice.
So that's where I thought this would go.
That's a better direction than I was heading on.
No, but I liked your direction. I liked your direction because I think there's an interplay here that's really important, where we get salinity, how it affects sensors and water content, how a toxicity question for the plants in the field. Certainly, this question of salt-affected soils, where we need a leaching fraction, right? And then this other concept that there could be enough salt in the soil to actually create stress a different way from matric. And we can't even measure that on the matric side. We can with the water potential or water content sensors. With the EC, we could estimate a salt stress, but we can't do it with the Terrace 21s. That was a fun one. I like that.
So here's another fun one then. We haven't had many questions come in about soilless media, so we're going to hit this one. So can you discuss how soilless organic media, such as biochar or cocoa coir, affects dielectric sensor accuracy? Does the physical shape and size of the pores holding the water being measured affect the measurement, since it affects the electrical path between the anode and cathode?
So every year when I teach this biophysics class, we have students who have to do class projects. And she did her class project on biochar, which is really interesting, and different amounts of amendment to soil. You'd never want to fully biochar something or other, right? It's only an additive to something else.
Right.
Turns into like this horrific mud.
It depends. It depends on what it's made from. Not all biochar is equal.
Oh, that's a good point. Hers did.
Okay.
She wrote me apologetic emails.
Oh,
It was
no.
yeah, no, she's awesome. So so how does it affect the dielectric? Actually, it doesn't. I don't
this
think,
one's easy to overthink it.
Yeah, that's what I think.
Because we're emitting an electromagnetic field into the soil, and it's going to kind of it's going to measure the charge time of a capacitor over the entire volume, right? So if you don't have anything conductive in there, and we know kind of where that limit is based off of our salt solutions.
And by the way, or anything else weird, but almost nobody has, can't remember, one of our friends had some really kind of strange soil from somewhere that had
you get like higher iron content or something in there.
Exactly.
Yeah,
it was really weird.
But that's extremely rare to get enough in there to
I've heard of one in more than 20 years, right?
Yeah.
So don't worry, everybody.
Right.
That's not a typical situation.
And so basically organic matter is going to change your calibration, not so much because of the electrical properties of what you're adding, but because of density, your interactive, your surface exchange capacities.
So as you mentioned, it would just be a function of bulk density, right? Below, I'm sure this is below 1
probably
gram per centimeter cube.
Yeah. So you might need a custom calibration for something like that, which is why we have soilless calibrations as well.
Yeah, and so physical shape and size of the pores, I don't think so. We're doing this electromagnetic measurements going over a volume.
Finer resolution than we can detect.
We got time for one, maybe two more. We'll see.
Okay.
We'll see how this goes. All right. What would be the best size container when measuring water content with different soils with a probe check?
The probe check doesn't really care. It's just going to give you a readout. But you should, for every sensor, and this isn't just meter sensors, you should take a look at what their measurement volumes are, how big of a soil volume, and then you'll want to at least get the minimum soil value. And I usually recommend a buffer because if you make a mistake there, then you can read air, which will bring your reading down, or things like that.
I agree. Yeah, just be conservative on your volume. Go a little higher than you think. I like that number 28, Brad. See, that may be a quick one.
Down to what water content percent? Can you trust the EC reading from the sensor?
Yeah.
The bulk EC is the bulk EC, so you can always trust that.
Yeah, but when's the last time you used bulk EC?
Actually, well, from a sensor, like I'm super sensor centric.
I mean, you've looked at it.
Yes.
But what did you do with it?
It's a great diagnostic tool.
I know.
But so if you're looking at water content, it's a corroborator for things that are happening in your water content. If you're trying to do some problem solving, and
but when's the last time you wrote?
I'm not going to be like, 0.3 bulk EC. Is this tap water or is this like
you're converting it, right? You're converting it to pore water. You see,
so a model is generally going to give you a better.
So the question here is can you use can you find a pore water EC at a water content of 10%? And the answer is no.
That's right, because that is too low. For the pore water EC, the bulk EC is still going to be good, but as Colin mentioned, that's not always that helpful.
So let me be clear on why this is true. We use a model called Hilhorst, Max Hilhorst. Real nice guy, met him years ago, just in a chance meeting in the Netherlands. And we talked over his model a little bit. The big thing is that it's in the denominator is your measurement of water content. It's the dielectric value. And as that gets lower and lower, if you're pushing below 20% water content, that essentially blows us up. It makes it huge. And so because it's in the denominator, because it actually subtracts some value, it just becomes really large. The denominator becomes small. The overall becomes really large, and there's big errors. So below 25%, I'd be really a little careful. Maybe 20%. What do you think? What's your experience?
Below 15%. Our software limit is 15%.
Is it 15?
We won't even give you a reading below 15% because it's not.
Yeah, if you're in Zentra Cloud, you'll think, the
Zentra Cloud,
your EC is broken.
Yeah,
and then we'll tell you, no, your EC is not broken.
And that's when you go to the bulk EC. Like, okay, the reading's fine.
Yeah.
Our water content conditions are just too low for this model to be helpful.
that's going to wrap it up for us today. So thank you all for joining us. We've had a really good crowd this whole time. This is our first time doing this, and it seems to have gone pretty well. I know that we enjoyed the discussion over here. Hopefully you did as well. Thank you again also for all of your great questions. We had a ton that came in via email before the session today. We had several come in, a bunch come in during this live Q&A. We tried to get to a bunch of them. Again, if we did not get to your question, we will be getting back to you, Colin or Chris or somebody else from our meter environment team, to answer your questions directly via email. Also, please consider answering the short survey that will appear after we finish here, just to tell us what types of Q&A themes you'd like to see in the future. Also, for more information on what you've heard from us today, please visit us at metergroup.com. Finally, look for the recording of today's presentation in your email, and stay tuned for future Meter Office Hours events. Thanks again. Stay safe. Have a great day.